thank Gully for inviting me to come, and it's been a pleasure meeting people and seeing old friends. Okay, so I'm going to talk about our efforts to use wikis for community annotation, and we've already heard a little bit of wiki skepticism, so we'll hope to dispel some of that for the future. Okay, so the basic problem, as we all know, is that everyone has their different version of this sort of figure of getting literature and data sets into the databases. And the key problem is that curators are rate limiting. And it was striking to see that Judy talk about there are people from MGI talking about the 26 curators at MGI, the 10 curators at Reactome. For most databases, the number is either 1 or 0 for a lot of organisms. So the solution that many people have come up with over the years to try and solve this problem of curators being rate limiting and expensive is to try and enlist the broader scientific community into participating in annotation. And of course, this has got a number of problems, and the most successful one example of a community annotation system is of course Wikipedia, where unpaid community has created more than 4 million articles just in the English version of Wikipedia, and then even almost a decade ago, it was shown that the quality of Wikipedia articles for science is actually comparable to or better than professionally curated articles. So the problem with wikis that most people think is that because anyone can edit, and in our case it's anyone who's registered can edit, that you don't know if the information is correct and you're concerned about this problem of chaos, that there's essentially little quality control because it's not, it's explicitly designed to not have a gatekeeper. It turns out that really isn't the real problem for wiki-based community annotation. The real problem was sort of highlighted in this Nature News article about BioWikis, in which I actually got interviewed and quoted, where I basically said that nobody's going to get tenure or their grant based on the fact that they edited my wiki. Okay, so what I forgot to say to the interviewer, and this is a really bad oversight, is that we were actually working on solving that problem. And we're not solving that problem for everyone, and we're probably not solving it for most of the people in this room. But we are solving it for people who work in institutions like mine. And our approach to that is to couple annotation to teaching, because we get credit for teaching, and NSF likes things that are connected to education and the broader impacts, and so on. So we've created this thing which we call the Community Assessment of Community Annotation Ontologies. Judy and Paul and all the people who go to Go Consortium meetings can fall asleep now because they've heard this all many times. The basic idea is that we're doing literature-based Go annotation, the kind of stuff you heard about that professional curators do. We're doing it as a team sport. Okay, it can be done intramural all the way to international. Our first time doing it was just inside Texas A&M. The second time we did it against the University College London. And it's all done on this publicly viewable wiki, which we call GoNuts, which stands for the Gene Ontology Normal Usage Tracking System. It's the wiki for Go that was there before the Go Consortium had their own wiki, but we had to name it something different because they wanted their own. Okay, so the basic idea behind CACAO is that first we train, give some really basic training to students about the general idea of annotation, what it's good for, the idea of gene ontology, and sort of the rules for CACAO. And we actually do this not just at Texas A&M, we do this at other participating institutions. And by we, a large part of this is done by my assistant research scientist, Brenly McIntosh, who I was Skyping back and forth in the previous talk trying to get data, last-minute data updates for my slides. And so Brenly often does remote training to other places via Skype. There's actually some videos embedded in our website that are her giving the spiel on how the CACAO works. And then we provide example papers for students to work with. And then, because A&M is on the semester system, we typically run what we call 5 innings of annotation per semester. And each inning has the top of the inning and the bottom of the inning. And the top of the inning is what we call the annotation period, and the bottom of the inning is what we call the challenge period. And then while this is all happening, we have ongoing assessment that's done by people who work for me. And now we've done it long enough, we have senior students who have gotten so good at the competition that we take them out so it's not unfair to the newbies, and they actually help with the assessment. Okay, so the basic idea in a CACAO inning is we have the annotation period. And the annotation period is pretty much like what you've heard the professional curators do. The students are now the curators. They've had some really basic training. They're not nearly as good as people like Harold and whoever else is in the room curating, but we restrict what they can work on. We essentially say there are only certain types of Go terms, there's certain types of Go terms that are problematic. We just say you can't use those. There's certain evidence codes that students have trouble with. We just say you can't use those either. So we try and steer them to the low-hanging fruit. One of the things we find is they actually try to bite off more than they can chew, much more often than they should, until they figure out that they're getting slammed in the competition. In our class, we let students find papers and proteins of anything they're interested in. So that can be things that are in some other class that they're taking. It can be things that they're related to the research lab that they're doing their project in. We've had students pick papers based on the drug target of the medication that their roommate was taking. And of course, being undergraduates at a large institution, we've had students pick genes that are involved in the, in contaminating breweries for, contamination in the beer brewing industry. Okay, so there's obviously, the kind of things we've heard about for publication triage. This sort of thing would be extremely useful for our students because they start out having no idea how to pick papers, and they sort of learned through the bad experience of finding things and having their mentors say, no, this is not suitable for Go, this is not suitable for Go, this is not suitable for Go. Okay, so once they identify papers or start with proteins, they go to Uniprot, find the appropriate entry for the protein. It was interesting to hear Jana talk about the problems of finding out which papers actually apply to mice, because we see this problem a lot, that the students will find a paper and then they actually can't figure out what entry is appropriate for that protein because the authors forget to tell you if they're working with the human protein or the mouse protein or the rat protein or whatever. Once they find the appropriate entry, we have this script that automatically creates gene pages within our wiki. Basically, it runs a web service that goes off to Uniprot and EBI, pulls out the basic gene information, pulls out the existing annotations that are out there, creates this gene page, and then we have an editing interface, which we call table edit. We use that instead of semantic tables because we didn't know about semantic MediaWiki when we started, and now we're sort of deep into the, we've already done our own thing. And so there's some natural language processing opportunities there in terms of tagging the publications that they've already found. Actually, some of the things I think that Paul was pointing out that what Sinomine could do is even without the natural language processing, the shared markup capability would potentially be useful for us there. Okay, so we make these tables so that they enter things in and they'll enter in a Go ID, and then it'll actually, nowadays it will tell them if they didn't enter it in the proper format or if it's not corresponding to an existing term. They enter a publication, and often they'll put in like a PMC ID instead of a PubMed ID. Now we've changed it so it does, it catches that. But I think having tools to try and have the markup shareable is good. And then an important thing is that one of the fields in the annotation they have to do, which is not part of the Go consortium standard, is they have to write text that says, here's the part of the paper where I think, which justifies this annotation. So they have to write, figure 2 shows an enzyme assay which justifies my assigning this molecular function term to to this protein. And one of the things that's a little bit different from most classes they take is that we tell them we're extremely brutal about partial credit. Essentially, the default is your annotation is wrong, and you have to convince us that it's right. And the reason why that works in a class is because they have an infinite number of opportunities to add more points by getting it right. Okay, so the students enter their annotations. The annotations go into this big scoreboard where they can view all the annotations made for a given inning, and then every other week we essentially say you can't add annotations, you can only do challenges. And then what happens in the challenges is they have this interface where they can click. There's no pointer, right? Well, actually, the video. They can click, they get this interface where they can enter their challenge. The challenge basically says, no, this is wrong because the Go term you used really should be this other Go term, which is more specific, less specific based on the evidence. You have the wrong evidence code, and so on. And I think that there's some NLP opportunities here that are a little bit different from what most people are seeing, because we'd actually like to be able to use the natural language processing to somehow compare. It's almost like the annotation agreement that, that Kevin talked about. We'd like to compare what the computer says with what the student said, with the assumption that both of them are sort of equally naive. Okay, while this is all going on, we have people who work for the project who are assessing and giving tips to the students. There's again a user interface that does assessments. This has been evolving over the course of the project. So now as you're assessing things, the assessor actually has to check off Did they get the right protein? Is it an appropriate paper? Did they get the right evidence code? And so on. And then there's a whole history of challenges and assessments that can be viewed for any given annotation. When we first did this, we basically had this idea that the first person to challenge an annotation would sort of lock the annotation to further challenges, and then we realized that people were putting in trivial challenges and the more interesting challenges were getting blocked off. So now we have sort of a one-to-many relationship where multiple challenges can be made, and it's sort of like whoever gets the right, whoever makes the annotation better first, but not necessarily the first one to challenge, gets the points. Okay, so we've been doing this for a while now, and this is sort of our latest updated information. Through last spring, we've had a total of 872 students participate in this, so that's a pretty decent community participation. They're distributed over a bunch of schools, which you'll see in the acknowledgements. And it varies from year to year, and basically the big fluctuations have to do with whether or not certain instructors who couple this to large classes participate or not. And so that's why I think like the big bump in fall of 2011 was when I think both Michigan State and Central Florida, I think it was, coupled CACAO to a large introductory microbiology class. And then as we go through the, review them and figure out which ones are acceptable, you can see that the fraction that is acceptable, is not necessarily that great, but it turns out because of that extra text markup that they have to do, it's fairly easy to find and reject the problematic annotations. So in the end, we have about, we've had 600, over 1,000, annotations that we're sending to back to UniProt through EBI. Many of these, interestingly, are from taxa that don't have monologism databases, or not ones that are going to be high priority. I think those are things that might be, especially be interesting for including in things like the phylogenetic annotation projects that Paul's involved in. Okay, so that's the basic overview of what we've been doing, and then now I have a whole bunch of slides of sort of discussion points. All right, so the first thing is related to the infrastructure. So, wikis are known for the collaborative editing features and the fact that anybody can participate. It's important to point out that they're also based on the idea of revision control systems. So they're probably based on things that are more SVN or CVS-like than Git, but certainly they have revision history, they have attribution, they have systems for merging changes and so on. So that's actually, I think, very useful, and that's something that's missing from a lot of sort of traditional biological database systems. Also, it's because it sort of follows this idea that Gali was talking about in his first talk about taking advantage of open source software development. Okay, so MediaWiki is the platform we use. Of course, it's not, well, it's written in PHP, which is sort of the, if Perl is the ugly stepchild, it's like, PHP is the black sheep of the ugly stepchildren or something. But although it's written in PHP, it is object-oriented, it is open source, there's a big development community, and an example of how useful that is that there was early on in this project, we wanted to have a functionality where instead of people having to constantly copy things from PubMed entries, we wanted to make wiki pages about publications based on information we could pull from PubMed. And I didn't know how to do that because I was just learning, object-oriented PHP, and I was learning the MediaWiki structure. And so I posted a question to their software development, their MediaWiki, administrators list saying, which of these hooks should I use and how should I do this? And somebody who I've never met chimed in and said, oh, that looks like fun. Let me write that for you. And he sent me the script, and I just included it. It's still used in our code. Is the this unpaid thing. We sent him a T-shirt in the end, and that was what, instead of our $150,000 a year programmer, it was somebody who just committed it through open source. And then we've extended in lots of ways. Wikis intrinsically have a problem of not being very structured, and obviously structured data is important for being able to do data mining. We use this thing that I actually wrote while I was on sabbatical with Lincoln Stein many years ago called Table Edit. The similar things could be done with Semantic MediaWiki, and now actually the Wikimedia Foundation is launching this project called Wikidata, which is based on the idea of the value of having coupling structured data types into MediaWiki. And in the future, we've actually talked about trying to merge Table Edit into the sort of the broader Semantic MediaWiki project. But using these extensions, we can change the infrastructure to, it's been a moving target as we've figured out, oh yeah, there's additional questions we want to ask about the process of how the students do the annotation. Like figuring out what kinds of mistakes they make a lot and how we can modify our documentation and training to, to sort of help them not make so many mistakes. That's stuff that comes from our ability to mine out what they're actually doing from the thousands of things that they submit and the basic challenges they do. Okay, so like I said, we're working with EBI to try and get things back into the system. That's one of the things I'm going to, talk to Paul about at some point because we're working that. Okay, then, so I think that the problem, so I said that the real problem for wikis is the problem of getting participation. I think we've mostly solved that. And we're now at the point where, because the assessment is rate limiting, we actually are not being as aggressive about recruiting new schools to participate in CACAO. Effectively the problem of keeping up with giving the students feedback, was something that almost killed Brenly last spring. Okay, so the real question is how can we improve quality, so we can take advantage of the data mining to then look at the kinds of errors that are made. So if we look, for example, okay, so I back up. So the students make these annotations and they're challenged. Okay, so at the end of the day, we wind up having different classifications. We have annotations that were just perfectly fine from the moment the student put them in. Okay, we have annotations that were not fine when the student put them in, but a challenge made them fine. And then we have annotations that we wouldn't put in, but they're close. The student, for example, found the right figure, but didn't use, found the right figure, made the right inference, but didn't have the right evidence code. And so this chart says of the ones that we marked as still requiring changes at the end of the CACAO competition, some, like of 108 of those that we sampled, some of them had formatting issues. 57 used the wrong Go term. It was either too specific, not specific enough, or but it was somewhere fixable based on the evidence that they showed. 30 had the wrong evidence code. And 38 of them, the problem was really not with the annotation. The problem was with how they justified the annotation. And that's something that really would not be rejected in a GAF because it's a field that wouldn't even be in the GAF. Okay. Then among the ones that were just completely rejected, there were about half of them were just things where the students didn't understand the science and tried to put in something that didn't make any sense. But there are other cases where it was incomplete, or they actually didn't notice that somebody had already, well, either they didn't notice that they were trying to put one over on us, that there were duplicates where there was some annotation that had already been made and was in the set that was coming out of Uniprot. Some mod or Uniprot curator had already made that annotation from the same paper with the same evidence code and the same data. Again, in some cases we don't allow annotations from review articles because it has to be evidence-based. So in some cases, students still try to justify, make annotations based on model figures from review articles and so on. 35 of them were this problem of just picking the wrong protein. And so figuring out how to get students to get the right Uniprot entry from papers, that turned out to be a bigger problem than we thought it would be at the very beginning. Okay, so how can we improve it? As I said, the fraction of problems is high. I think that natural language processing could help us in both quantity and quality by making it more efficient in all the same ways that apply to professional curators in terms of paper triage, tagging, and so on. But I think the thing that's a little bit different about what we do is that we could actually also use NLP processing of the actual text that the students are putting in to justify the annotations, to try and classify it into we would like to classify the annotation activities so we can understand more what their processes are, because they're not necessarily following the workflows that we dictate in the way that you would if it's somebody who's working for you as a paid curator. Okay, the other thing I should point out is that we call this the community assessment of community annotation with ontologies, and I'll admit partly that's because it was a cute acronym and we sort of retronymed it, but it really isn't a community assessment yet. And I think we've now got to the point where we have the infrastructure to create a real community assessment where we would do things like CASP or CAFA or the other competitions that are out there, for example, where we would have blinded papers corpora of papers that were curated by professionals that were not, where the annotations were not put into the into the public release yet, and then we could actually steer the students towards those papers and see, do they find the same things? You know, what I expect would happen is that in some subset they'll find things. The students tend to be not very good about finding all the annotations that you can get out of a paper, okay? But we also are set up where we can test hypotheses, like would we do better if we changed the point structure so that you got more points for a challenge than you got for an annotation? That, you know, because part of the whole hypothesis here is that proofreading, you know, the whole wiki notion is that it's like biology. Proofreading gives you higher fidelity, and so if we don't get enough annotations being challenged right now, can we make it easier? And in fact, some of the ways in which the natural language processing could help is by helping the students and the assessors identify those annotations that are potentially problematic, that could be sources of challenge points. Because what we find is that the students who win each year, the teams that win, usually win on challenges, not on their raw annotations. They tend to have the most, they find the most problems with other people's stuff. You know, and then if we actually had massive more resources than we do, we could do Google-like things like see if it matters whether or not two different UIs work with different, you know, different competitions, like do the colors matter, do the fonts matter, stuff like that. And so there's a ton of things that we could do in terms of assessing it, in terms of the efficiency for annotation. Now, a lot of this course requires, you know, the big variable is that different students are being involved every semester, different instructors. That's a bigger you know, it's a bigger variable than anything that we would vary. So we'd like to be able to scale this stuff, and that's where, again, help with software development would help, and help with community involvement with the assessment would help, is that if we can scale this further, then we can actually get real data on how these variables affect the efficiency of annotation. And the last thing I wanted to point out is that we're really interested in assessing, you know, the quality, you know, what we can do to, from the point of view of the interest of the mods and the databases, of how we can get community annotation to work for curation. Because we're coupling it to teaching, there's a whole different kind of assessment that's related to the educational value of what we're doing. And sometimes those interests are not necessarily in alignment. Okay, so for example, for the educational assessment, some people have advised us, like, do this on a smaller scale with fewer students and fewer annotations. And it's like, I'm not sure if I'm interested in that, but we have this platform now where people could do that if they wanted to. Okay, so the last thing is just the extensive acknowledgements. You know, Brenly, as I said, does most of the work. The original software that modified MediaWiki was done by Daniel, and Brenly and Daniel just got married last week. Susie Alexander actually has been to a Go Consortium meeting, and she started out as a CACAO student, now works with us to actually help run the whole thing. Okay, it's funded by Portico, which Paul's the PI. We've had lots of good feedback from the Go Consortium and other people. There's 872 students over the course of doing this, lots of schools. The green ones are basically because we just recently did a plant-specific collaboration, CACAO, where, alas, my alma mater, Stanford, had its ass kicked by UC Davis. And so it's funded as head through the Portico project, and basically thank all of them, and thank you for your attention. Okay. Yeah.
So any questions? Lots of questions. Yolanda, and then William.
Yeah, this is fantastic work. I really enjoyed it. We're doing a lot of work with semantic media wiki for scientists, particularly in the geosciences, so I'd be very interested to talk to you about this. My question is about how you approach using the wiki mechanism for this. Typically, wiki repositories work very well because you view them as a social technical system. So one side of it is the wiki and the interface of it to the users, but the other side is the non-technical side of the wiki, which is the social side. So things like Wikipedia and many other successful wikis, they have social rules for how the community needs to interact together, and in Wikipedia is very, very elaborate. Mm-hmm. So things like promoting some of your editors to oversee others, and having different ranks in the editing scale based on skills or capabilities or seniority and so on. So I wonder if, you know, there may be opportunities to introduce these more social aspects of wiki communities into the courses and the activities that you do.
I think that's a great question. It's something that we haven't followed in Wikipedia's footsteps so much, partly because a lot of those things evolved to deal with the fact that they had a large enough community that they actually start getting edit conflicts. right? And we're still at that level where there's so few people editing that there's very few conflicts to resolve. But the students, even though we don't have the real formal educational assessments, the students get really excited by it. And a lot of the students we have are doing it are students who repeat it. And we even had students who've participated, just as auditors and not for credit because they just thought it was cool and they want to take it off and set it up when they go to graduate school and so on. So those people sort of informally become promoted in these ways that you've been talking about. And I think some ways to actually recognize that on their user pages or within the Wiki community, I think would be, there are things we've talked about we haven't implemented yet. Like we've talked about having some sort of badge that you could even put on your own website. This is what this automatically shows you my contributions to the wikis that we've done. But I'd be interested to hear about other ideas to do that.
Yeah, that's kind of to follow up on that. Gamification is the thing that everyone always goes to when they think of how do we get more participation and better quality and assessment and everything. But it's definitely a lot more easier said than done. But there's some good examples out there. We can talk more about that. My question, which is probably the last thing you want to hear if you're already kind of at a rate limiting step with assessment, is a lot of the big crowdsourcing projects that have been that have done really, really well, Polymath and Galaxy Zoo, they have not focused on the research article as their primary object, but rather a research finding. And so, like, as more scholarship moves away from PDFs and moves onto blogs, how are you going to, and other places like that, how are you going to incorporate, or do you plan to incorporate annotations of those as evidence, as well as the traditional published research, the literature, as the boundaries kind of get fuzzier? How do you think you can adapt with that?
So right now, I'd say that within what we're doing as ontology-based curation, that's sort of a question more for the Go PIs, right? Is in terms of how one does. One of the requirements is that for certain evidence codes, you require this to really be connected to the literature, and that's considered part of being gold standard for reliability. I think that there may be some interesting non-traceable author statement codes here. I don't know if that's really been talked about that much in terms of how one would deal with it. Judy's squirming a little bit. You want to say anything or no? Okay, yeah, that could be a good post-dinner discussion with alcohol. Yeah.
Yeah, so one of the things that I think is interesting about this is it introduces a different triage step, right? So you have triage not only at the level of which articles are you even going to curate, but also really you have a second level of review or triage, right? Where now someone's gone through and made some tentative annotations or some putative annotations that you may want to accept. I guess if you're trying to look though at how useful that would be to say incorporation by model organism databases and really becoming the corpus of knowledge represented there, how would you one of the things you've looked at really well, I think, is the error rates of annotations that you have. But what about missing annotations? In other words, do the students annotate the same way a curator would, or do they miss a lot as well? And I know that's a harder thing to assess because you actually have to ask the assessors to go back and read the entire papers, but I think it's a useful thing to do because you really need to know what you're missing before you would then say, well, I could use some crowdsourcing here, and then expect that at least they put in enough of the wheat, right, that I can go in and reliably use that as a step for these processes.
Right, so I think it's clear that the students don't annotate the same way that the professional curators do, especially in that area of extracting the number, the number of annotations they extract per paper is low. I was actually just trying to figure out how to plot that, during the break. But when I look at the number of annotations per paper, there's a really long tail of singletons, right? Or the number of annotations per protein, there's a really long tail of singletons. Harold.
So I guess, just follow up though. Could you put that into your scoring scheme as well? Right, if you think that's part of the problem. Anyway, it's just something to think about.
Right, I mean, we actually have thought about it. I'm not sure exactly how to know this is again one of these things like what's the right answer. So it may be that the paper actually really only has one thing suitable for Go, and so you don't necessarily want to give, but yeah, there may be ways. Again, this is what I was talking about, manipulating the scoring scheme. Is could it be that you get extra points for finding something that somebody missed in the same paper, for example. That could probably be done. It's non-trivial coding. Again, this is also where the stuff that Gully was talking about, crowdsourcing, the actual infrastructure part would be interesting as well.
Yeah, so I was just wondering, do you have any feel for, if you look at the number of successful annotations or correct annotations versus ones that you had to reject, with the level of the students that made the annotation and maybe the background courses that those students have or have not taken?
That's the kind of stuff that so one of the people who's on the acknowledgement slide is Karen Serum from Bowling Green, and she's actually an educational assessment expert, and so that's the kind of thing we want to work with Karen to do better. There's issues of like, what year they are, how much background. There's also like, we think anecdotally, although that they get better from inning to inning. So the fifth inning annotations tend to be better than the second inning annotations. The first inning annotations tend to be pretty good because we do lots of hand holding in the mentoring during the first inning. And then it tends to drop off and come back up. But that's probably not rigorously analyzed as well as it should be. But yeah, all those sorts of questions I think are things that we want to be able to pull out of the system. We just don't have enough.
So the last question before lunch.
It's actually a suggestion. So for this biocreative that I will talk later this afternoon, there is a Go corpus. So maybe that could be used to provide the students examples on how to improve their annotation, because in that corpus you have all the sentences that give evidence for a certain Go term in the full text, and is extensively annotated along with the evidence code. So I think that could be either used as a test set for you to see how much they are missing if they want to try, or you can use it as an examples for them to see how annotation.
But so are these generated as being?
These were manually generated by curators from TARE, MaceGDB.
But are they held back from the public annotations then, or?
No. These are new papers that are being annotated, and right now there's annotation of 150, and there will be 50 more for the test set, which we do not, is a blind set, so we're not gonna expose the Go annotations yet. We're gonna hold for them, but I think that would be a good set for you.
Yeah, I mean the blind set especially would be useful, because one of the things that when we create the gene page, we suck everything that's already in QuickGo. Right, so if it's already there.
But the thing is, for this set, the blind set, I think it will be held until October, because after that, it's the biocreative workshop, so it will be made public. So I don't know.
Yeah, no, I've been trying to think about ways in which we could couple with biocreative. That would be good to talk to you about that.
Great. So thanks again for a fantastic talk. Let's show some appreciation for the whole session. Thank you, everybody who presented, and thanks for the good questions.